Hello and welcome to the pre-lab lecture video for the chromatography experiment. I am just going to pull up my slides. All right, here we go. So as I mentioned, we're going into the last experiment, which is the chromatography experiment. And we're going to be talking about the general theory and the experiment as well. So the theory for chromatography is that it's a way of separating compounds in a mixture based on its distribution between two phases. And typically that would be a liquid or mobile phase and a solid, also known as a stationary phase. But like we'll talk about at the end of the video, it's possible to have, for example, a liquid stationary phase and a gas mobile phase in a different type of chromatography. So again, chromatography is a way to separate compounds or purify compounds. And that's really important because oftentimes organic chemists get a mixture of compounds and they want to purify or separate those compounds. And so chromatography is one of our methods for being able to do that. You guys have learned other methods previously, for example, distillation, which was used to separate mixtures of both miscible and immiscible liquids. You've also learned about extraction, which is a way to separate compounds based on their affinity for an aqueous or an organic phase. And we talked about acid-base extraction, which is one main type of liquid-liquid extraction that's used in organic chemistry. So there are many ways to separate, and one really important way is to do chromatography. So we mentioned that there's two phases and that we can separate compounds based on their affinity for those two phases. Let's talk about those phases in a little bit more detail. So we have this stationary phase, and again, typically it's a solid and remains fixed in place. So the most common solid that's used is silica gel, but silica gel is a little bit acidic, and so for certain very acid-sensitive compounds, they're all alternative options. For example, alumina, which is neutral and so does not cause compounds that are acid sensitive to react when we don't want them to. And there's also a mobile phase, also known as an eluent, and that mobile phase will be moving through the stationary phase. And again, based on a compound's affinity for either the stationary phase or the mobile phase, the compound will move at a different rate, thereby separating it from other compounds in the mixture. So typically our mobile phase is an organic solvent, and it could be common solvents such as ethyl acetate, hexane, dichloromethane, and methanol. One of the reasons why those four solvents in particular are so common is because of the fact that ethyl acetate and hexane are miscible. Even though ethyl acetate is much more polar than hexane, they do happen to be miscible, which is a great feature for making different polarity solvents by varying the amount of ethyl acetate or hexane in a mixture. And then also dichloromethane and methanol are a miscible pair of solvents. Dichloromethane is not as polar as methanol, but they still mix in all proportions, which is great. So we can vary or get a different mixtures, maybe 50-50, 80-20, that sort of thing, which have different polarities based on the amount of dichloromethane or methanol, for example, in the mixture. So this is a picture here of a TLC plate. It's kind of just a not realistic, but sort of a graphic visual of the process. So in TLC, which stands for thin layer chromatography, we have a plate which has silica gel powder on one side of it. Compounds are spotted onto the plate at the bottom, and then an eluent or an organic solvent is used, which will move up the plate through capillary action. And based on the polarity of the compound and its interaction with silica gel versus the solvent, each compound would hopefully move at a different rate, allowing for separation. Column works a little bit differently. Silica gel is put into a cylindrical column, and solvent is run through it from top to bottom, which is different from TLC, where solvent runs from bottom to top. And then compounds either adhere very strongly to silica gel and move very slowly, or they don't adhere to silica gel very well, and so they'll move much more quickly through the column and then separating that way. So again, compounds sort of move out the bottom of a column. And let's say if it was the red compound, so it is a compound that's moved highest up the plate. That one would actually move the fastest through the column and be down towards the bottom of the column. But again, we'll talk about those differences between these two different types of chromatographic techniques as we move through the theory. First, let's talk a little bit more about silica gel. It is a white powdery substance, and the structure is a series of silicon and oxygen atoms bonded together. And on the outskirts of the rather large structure, solid structure, are OH groups. So for that reason, we say silica gel is really polar. Look at all of these OH groups. So it tends to interact really well with polar compounds and also those that can hydrogen bond, and not so much nonpolar compounds, particularly ones that can't hydrogen bond. And when we're doing TLC, again, that stands for thin layer chromatography, we have a plate that could be made of plastic or glass. And then on one side, it's coated with the silica gel powder. And then a column, again, has a mixture of the silica gel powder mixed with solvent, and it forms a uniform, homogeneous mixture that the compounds can then pass through as they interact with the silica gel and the mobile phase. And because we always want to collect any solvent that might come out of the bottom, you can see this beaker here collecting that solvent. It is important that we take note of an important safety feature of silica gel. It is very hazardous when inhaled. And so we want to try to use that in the hood, not outside the hood, with the sash down as much as possible to avoid, inhaling the silica gel, because it can be hazardous in your lungs. Okay, so let's talk about compounds with different functional groups, right? So how well will they bind on average with silica gel? So if you notice, the compounds that have the strongest affinity for silica gel are compounds that not only can hydrogen bond, but are very polar overall with oxygen and nitrogen atoms. And then moving on to compounds that don't hydrogen bond, but still have oxygen and therefore are fairly moderately polar. And then our non-polar compounds like alkenes and alkynes and alkyl halides will not adhere very well to silica gel. They have the weakest interaction with it, and therefore they're likely to pass right through the silica gel and not be strongly bound to it. And so they'll move really quickly as opposed to slowly through a column or up a TLC. So what if we have a compound and it's adhering really strongly to silica gel because it's pretty polar, but we want it to move through that column or move up the plate so that we can see separation from other components in the mixture? Well, we can control how quickly these compounds move through silica gel by choosing the correct eluent, right? So we want to note that, again, eluents or the mobile phase for chromatography are typically organic solvents with a low boiling point that are fairly inert towards reactivity. And so we would choose, right, based on polarity. So if you have, for example, if you use an alkane, then that's only really going to attract nonpolar molecules, and it's not going to have much affinity for polar molecules, and therefore it's unlikely to get polar molecules to separate the interactions between silica gel and the molecules, and to get those molecules moving through the silica gel. Okay, so if you want those polar compounds to move, we need to choose a more polar solvent, right? So you can see here the allotropic series for silica gel. So for example, if pentane isn't working to get compounds moving on that plate or down that column, we could choose more polar solvents such as diethyl ether, ethyl acetate, ethanol, acetone, and all the way up to acetic acid and methanol, right, which will bind to polar compounds, disrupt the interactions of those compounds with silica gel, and get those compounds moving right along through that silica gel. So we can control how quickly compounds move just by choosing a different solvent. Now, here's a little bit of a tricky thing that students don't get typically on the first go around when they're learning about chromatography. If you have compounds that have separated on a plate, and then you, for example, try those compounds on a different plate using a different eluent that's more polar, sometimes people think, well, only the polar compounds will move more, not the non-polar compounds. But actually, all of the spots, all of the compounds on the plate will move more quickly with a more polar solvent. Even the non-polar compounds, because again, they're not really adhering to silica gel very well in the first place. So if you've got a bunch of spots that are kind of hovering towards the bottom of a TLC plate, or stuck on the top of a column, and you want to get them to start moving, you need to use a more polar solvent, and that will get all of the compounds moving more quickly, and still relatively in their order based on their affinity for the silica gel in the first place. Okay, so what are these techniques useful for? That's something important to think about. So TLC is useful for small-scale qualitative analysis. So that it specifically says qualitative analysis because even though we're able to potentially visualize that there's more than one component in a mixture, we're not able to really quantitatively say how much is present of each compound. So for example, through TLC, you might separate a mixture and say, oh my gosh, there's 5 compounds in this mixture. But there's no way to know that, there's 50% of one of the compounds, 20% of the other, and 10% of the third, and so on, right? So we don't have any way to quantitatively tell you how much of each compound is in a mixture. But we can say, well, there's 5 components in this mixture because we see 5 different spots on the TLC plate. So we can tell, right, if there's a mixture, but it's not a quantitative analysis. Okay, some of the great things about TLC, you only need a very, very small amount, right? Less than 1 milligram dissolved in the solvent is plenty of compound to spot on a plate. We're only just putting the tiniest amount on the very small TLC plate. So you don't need much, which is great, because sometimes you don't have very much of a compound. And you don't get it back. So once it's on the plate, it's waste, essentially. So there are ways on a larger scale to do it, to get it back, but generally it's just wasted out. So it's good that you don't need a lot. And you can get information on the purity of compounds or mixtures rapidly. So the entire process to do the development of the TLC plate is only a few minutes. right? So it takes just a couple of minutes maybe to spot the plate, and then to put the plate into a development chamber, it takes 2 minutes, and then you can immediately analyze mixture. So it's very, very quick, and that's a definite advantage. If you have a standard sample, you can establish that two compounds are identical by looking at various TLC plates with different eluents, and if they always are migrating to the exact same amount, then it's very likely that they're identical. And it can aid in the analysis of progress of a chemical reaction. So we haven't done this yet where we've analyzed our chemical reactions. In the last reactions experiment, you just did your very first reaction where you calculated percent yield, but we didn't really monitor the progress. We just sort of, figured it would work fairly quickly and it would be purified through the distillation process. But not all organic reactions occur quickly, right? Some of them occur over several hours, some of them occur, over days. And so we want to be able to monitor the progress of a reaction fairly easily and quickly over time. So you could, for example, look and see, okay, after 10 minutes, what's the progress of this reaction? How much starting material is left, relatively speaking? Again, we don't know exact numbers. And then are we starting to see a product spot develop, right? And we're looking for the disappearance of the reactant, the limiting reactant spot, and the appearance of a product spot, and to indicate that a reaction has occurred. So we can look at that for progress over time, especially for slower reactions, right, that are occurring over several hours. We should be able to see it gradually. The starting material is converting more and more to product until there's only product left and the reactant is gone. You can also monitor the effectiveness of extractions with TLC and also column chromatographic separation. So we're going to talk about column chromatography even more. But after we've done this column and we've collected a bunch of fractions and we want to see, okay, did we actually separate the compounds on the column? And TLC will be our way of looking and seeing what exactly is in each fraction. So it's extremely useful, and organic chemists who are doing reactions are using TLC, all day, every day. Okay, what about column chromatography, right? What is it useful for? Well, column chromatography is useful for the separation or purification of compounds on a larger scale. So TLC just kind of gives you a mirror into what's in the mixture, right? But doesn't really actually help you, purify grams of material. But column chromatography can be used up to, many, many grams of material. We can actually help purify it from other components. It's going to be based on whether or not it will separate based on its affinity with silica gel. So you do need to have, compounds that have a different enough polarity, which correlates with affinity to silica gel, in order to have separation. But it, is able to separate large quantities of material, and the compounds can be liquids or solids. So you could separate a mixture of two liquids as long as they have a little bit of difference in their polarity, and which means difference in their affinity with silica gel, or two solids or a liquid and a solid. So it's not really specific to one or the other, for example, like distillation, which is more specific to liquids. Okay, so let's go ahead and look at the overall synthetic process. I thought it was important to do this because now we're on experiment 5, the final experiment, and it's important that we kind of understand from an organic chemist perspective, if they're a synthetic organic chemist and doing synthesis of different compounds, what does the overall process look like with all of the different techniques that we've studied up to this point? So we're going to start up here in our top left, right? And the first thing we would do typically is run a reaction, right? If we want to convert one compound to something else, then typically we would need to dissolve it in a solvent, add the reactants together, maybe heat it up, and facilitate the reaction from taking place. And we would be monitoring that reaction using TLC. This is a TLC plate. You can see that there's different spots at the bottom, and there's different components in each lane, right? So this lane only contains this less polar yellow spot, and then the second lane only contains the more polar orange spot. And then this third lane, right, is a mixture of the two. So maybe this is the reaction mixture, and these two compounds are standard. So perhaps they have a standard sample of the reactant and a standard sample of the product of the reaction. So they're running those side by side with the reaction mixture to see when the reaction mixture the reaction itself might be complete, right? So if this was the starting material, this was the product, we would hopefully see that this is gone and that at the end of the reaction, this is the only compound that remains. But it doesn't always work perfectly where you don't have any starting material at the end of a reaction, right? Some reactions are subject to equilibrium, where it's not ever going to go all the way to completion, and you're stuck with a mixture at the end. Okay, so let's say we're in the situation where we're stuck with a mixture at the end, or perhaps you have excess of a different reagent that's not the limiting reactant that's still present at the end in your reaction mixture with your desired product. So we've got this mixture, and now we need to separate the mixture when we deem the reaction to be complete. So typically the first purification or separation process that would be done is an extraction, right? So an extraction, again, uses an organic solvent and an aqueous layer. And sometimes acid-base extraction is done, or sometimes we just have reagents that would be more soluble in water than an organic solvent. And so we're able to start purifying the mixture by removing some compounds into the aqueous layer that are not our desired product. So we can do our extraction, hopefully remove some components from the reaction mixture in that process. And then again, once we're done with the extraction, we would take the organic layer and we would dry it, because some water will inevitably get into that organic layer during the extraction process. We would use some magnesium sulfate or other suitable drying agent to remove those trace amounts of water. We would then decant that mixture or filter it into a round bottom flask, right? Being careful not to fill it more than halfway, and use the rotary evaporator to remove the solvent, right, the organic solvent, and leave the compound of interest at the end. So typically we would be left with either an oil or a solid at the end of the extractive process. So let's say, unfortunately, we still have a mixture even after the extraction process has been done. We removed what we can, but not all of the impurities. So now we would further purify this liquid mixture, in this case, using column chromatography. And we'll see pictures that show it a little bit better, but this is a column filled with silica gel and solvents, and they're collecting fractions, right, in order to hopefully separate these two components, again, based on their different affinity for silica gel and the eluent that's being used as well. So after they've collected all of these fractions, we want to know what exactly is in these fractions, what compounds, right, between these two are in these fractions. Now these are colorful compounds, and so it might be easy to see where the compounds are because you can see which fractions are colored. But a lot of the times in the lab, compounds are not colored, or most organic compounds are not colored. And so we can visualize the plate using different methods other than just our eyes. And if they weren't colored, all of these fractions would be clear, right? Because the white solid would dissolve in the solvent and it would be a colorless solution. So we wouldn't really know where the compounds are, or have any idea if we can't see them. Then we would, so if we needed to figure out what's in these, right, we would have to do again another series of TLC plates. Now we're looking at every single fraction gets spotted onto a series of TLC plates, and then we see what's in those fractions. You can see what they've done here is always run a standard, which is the original mixture that they used and tried to separate using column chromatography. And then they have all of the fractions basically from fraction number 1 all the way to the end here. And then you can see that a really nonpolar compound came out in the first couple of fractions. That's kind of a surprise because we didn't even see that in the original mixture. And that can be the case. If there's just tiny amounts of it, sometimes you don't see it until it's a little bit more concentrated. And then this second, more larger spot starts getting eluted over several fractions, right? And it continues to get eluted over several more fractions. You might see, oh, there's like a little dot here, like what is that, right? This could just be like a compound the plate was contaminated with before you even ran it. So because there's nothing over here, it's really unlikely that there's anything in there. It's probably just a little false positive. So we just keep going, right, until you start to see, oh, there's some fractions that now have a mixture. Both components are in there. Those, unfortunately, we did not achieve separation because those fractions have 2 components in them. And then once that comes out, now we start to see fractions that only have the second, more polar compound in them and are fairly pure. So this is actually a fairly successful separation via column chromatography, where we have lots of fractions with the pure, less polar spot, and then several fractions with the pure, more polar spot. And just a couple of fractions that we would either discard or potentially collect and repurify using another column. So let's say we then, the compound of interest was that first less polar spot. We would combine all of those fractions, right, and into one giant round bottom flask and use the rotary evaporator again to remove the solvent. And then hopefully we would have pure compound, right, after that separation. So you can see how integral in the synthetic process the TLC and the column was to monitor the reaction and to monitor the purification process after the reaction. Once we have our pure compound, or what looks to be a pure compound by TLC, because there's only one spot, then we would do all of our analysis of that compound. So for example, we could get an NMR spectrum using a portion of the solid mixed with solvent, and we could get an IR spectrum, and then we could even get a GC or an LC, which are different types of chromatography we'll talk about at the end, that will actually give us quantitative purity of that component. You can see like this one looks like there might be a second impurity here and then one main compound, that mixture. So that would actually quantify for us how pure is this compound. And these two tell us, is it the compound that we're looking for, which is really important. And once we have that, then we can work on converting this to something else, right? And continuing that synthetic process. So that's sort of the overall synthetic process and how extraction and chromatography and running reactions and things fit into that process. Now let's look at each technique, TLC and column chromatography, a little bit more closely. And then we'll talk about exactly what we're doing in each part of the experiment. And at the very end, I'll talk briefly about gas chromatography. Okay, so let's look at thin layer chromatography, also known as TLC, first. So this is a method where it says purification, but really I probably should have said separation of low to moderately polar compounds. It says that because if the compounds are too polar, they actually adhere so strongly to silica gel that they likely won't um, move off of the spot that you put on the plate to start with, and therefore we can't separate them from other components, right, if they refuse to budge because they're adhered so strongly with silica gel because they're so polar. So in that case, there are other methods you might have read about in the technique manual called like reverse phase, TLC, and things like that, that can be used to purify more polar components. But typically, most organic compounds are not that polar, right? They're sort of in the low to moderate range. So this silica gel is the perfect absorbent for that. So again, the silica gel is spread into a very thin layer on either a glass or plastic backing or plate. And the mixture to be separated is dissolved in a little bit of solvent. That's to make sure it's not too concentrated. If we were to put a pure compound on the plate, it would be so much compound that it would just streak all over the plate and wouldn't give us the visualization of the separation that we're looking for. So again, we dissolve it into a little bit of solvent that will evaporate anyway, so it's not going to affect the TLC process. And then we will use a capillary tube to spot that compound that's dissolved in solvent onto the bottom of the plate after we draw a pencil line. And I'll talk more about that in a minute. And then we will use a developing chamber in order to develop the plate. And inside the developing chamber, we'll put the solvent or eluent. We will put it in the bottom of the plate. And again, it will move up the TLC plate through capillary action, moving the components hopefully right along with it. This is actually a piece of filter paper that you might use commonly with like a Buchner funnel. We can actually use this as well, fold it or cut it, place it in the plate, in the developing chamber, and it helps to keep the entire air above the solvent sort of saturated with solvent and not dry, so that the plate, as the solvent moves up the plate doesn't dry out. And then again, we will need a pencil. So these are the supplies you will need to do a TLC. Okay, so let's look at that process in even more detail. So again, we will need to acquire a TLC plate. It might have a glass backing or it might have a plastic backing. And on one side of that plate, there will be a layer of silica gel. You can see there will be a layer of a solid white substance. The other side will just be glass, right? So we want to make sure as we're spotting compounds onto a plate, we spot them onto the side with the silica gel, not onto the side that's just glass. It's not going to absorb the compound at all. So we would take this plate and we'll use a pencil. So pens have organic inks in them that can move along with the plate. So we would not want to use a pen with ink in it. We would just want to use a pencil, which is just made of graphite. And we'll draw a line, a straight line on the bottom of the plate. And most people use a ruler to make sure the line is really straight, unless you're very good at freehanding a straight line. And that line that we draw with pencil is typically 1 to 2 centimeters from the bottom of the plate. And then you can even put little dots with your pencil where you plan to spot compound. That you know, okay, there's 3 dots, I'm going to spot 3 different mixtures onto this plate, and this is exactly where I place them. Because the compounds will move up the plate, but the pencil mark will still be there at the end. So again, you're going to use these capillary tubes. The capillary tubes are not the same ones that we used for melting point analysis. ones for melting point analysis have a closed end, right, for the solid to go into. But the capillary tubes are actually open on both ends. And that's important so that the compound will go into the capillary tube and then through pressure come out the bottom of the capillary tube. And so you will dip that capillary tube into the compound that's again been dissolved in some sort of organic solvent, and then spot that dissolved mixture onto somewhere on the plate on the bottom here. And as you just touch it to the silica gel, some of the compound will come out of the plate. And if you just leave it there, eventually all of the compound will come out of the plate. You'll get this really big dot. That's not what you're going for. You want a relatively small dot so that as it moves up the plate, you have small dots moving up the plate and not giant dots. So you just want it to, to touch it, allow some to come out, but keep the spot fairly small. And so move the capillary tube away. And then the solvent, because it has a very low boiling point, will evaporate. And it'll just leave the compound on the plate. So the solvent is kind of just a vehicle to get it onto the plate in a really dilute fashion. But it doesn't affect the separation because you're going to allow it to evaporate before you develop the TLC plate. If you're going to use the same capillary tube to spot multiple different mixtures, it's important that you clean the capillary tube in between. You can do that with just some acetone. And so once this plate has been appropriately spotted, if I can't see the compounds because they're not colored, then what I would usually do is take it over to an ultraviolet light chamber. And I'll show you that when we get into the lab so that I can see the compounds, right, or the spots on the bottom of the plate before I even develop the plate. Because sometimes the mixture that you place onto the plate, the dissolved mixture, is too dilute. Very little compound gets onto the plate, and it's mostly solvent that you're placing onto the plate. If that's the case and you can't even see the spot before you develop the plate, you're never going to see the spot after you develop the plate. So you're going to waste your time developing a plate that doesn't have enough compound on it to even be able to visualize spots at the end of the process. You always want to kind of double check, okay, do I see a spot at the bottom of the plate everywhere there's supposed to be one before I develop the plate? Because there's ways to add more if you don't see a spot. So let's say you don't see a spot here, or it's very, very light on one of them. What you can do is go get more of that mixture and spot it right on top of that other spot. Allow the solvent to evaporate. Place more, right? Allow the solvent to evaporate. Place more, and you can, do that as many times as you want to get more compound onto the plate. So it's always good to check before you bother developing the plate. To develop the plate, we're going to place it into what's called a developing chamber. This one has just like a plastic wrap for a top, and this one has a black lid, probably. Either one is sufficient. And you can see this is the filter paper that's used in the TLC plate. And again, once we get in the lab, I'll show you how to do that effectively. And it's kind of sat or curved around the glass inside. We're going to add the eluent into the plate. It is or into the developing chamber. It is really important that the eluent does not go above the line where the spots are, right? Because if it does that, all it's going to do is dissolve up these compounds and they'll stay down here and they won't go up the plate. So that's bad. So this amount of eluent in the bottom, as long as it's below the starting line for the spots, then you're good to go. And hopefully it gets a nice flat layer too. So in our developing chambers, unfortunately, they're kind of curved on the bottom. So we do need to add a fair amount of solvent in there in order to make sure that the solvent is flat on the top. Because if it's not flat on the top, then the solvent won't form a flat line on the bottom of the plate, and it won't move the spots sort of uniformly up the plate. But those are kind of nuances that we'll talk about as we get into the lab. Okay, so at some point, we're going to take the plate out of the developing chamber. When do we know that we want to do that? So basically, we want the solvent to get fairly close to the top of the plate without going to the very, very top of the plate. So once it's the solvent front is sort of really close to the top of the plate, we can take the plate out, and then we can mark the solvent front with a pencil. And that's going to be important that we mark that line. But even if you forget, it's okay, because oftentimes you can sort of see the solvent front as you're looking at the plate under the UV light. Okay, so we talked before about the fact that some compounds are just colored, and you can see them on the plate because they're colored. But most organic compounds are not colored. They are white solids or colorless liquids, and so you can't see it on the plate at all. So how do we then visualize the plate so we can actually see the compounds in the different spots on the plate? We need a different method for visualization for those compounds that are not colored. A common visualization method is using ultraviolet light. That works for compounds that are sufficiently unsaturated. They usually have at least one aromatic ring or several what we call conjugated pi bonds, where the pi bonds basically are separated by sigma bonds, just one sigma bond in between each pi bond. Those are conjugated pi bonds. As long as there's enough of those, you can see the compounds using ultraviolet light. So this is, for example, what it might look like. The plate itself kind of glows green and the spots are differentiated. We can see like this lane just has one spot, another spot looks totally different, kind of a blue, and then the spot all the way that's moving along with the solvent front. Very, very non-polar compound. And then a mixture of all three. If the compound doesn't have sufficient unsaturation, then you might need to use a different method for visualizing the spots. Another common way is to use what we call an iodine chamber. And an iodine chamber is just a solid iodine inside of a container. And the iodine sublimes and fills the chamber with iodine gas, and that'll adhere to the spots on the plate. So sometimes that'll visualize it. Alternately, some people mix iodine with silica gel to make a different type of iodine chamber. So both of those work. And then if that doesn't work to visualize compounds, because maybe iodine doesn't adhere to certain compounds, there's a variety of different types of what we call stains, chemical stains, using different compounds like potassium permanganate or vanillin, a bunch or mixtures of different compounds that actually chemically react with the compounds, and then will hopefully show spots after that analysis takes place. Sometimes it'll just react at room temperature with those stains. Sometimes you have to heat it up. So there's a variety of methods you could use if you're struggling to visualize compounds. But you won't struggle in this lab because we've made sure that the compounds are nicely unsaturated and will either be colored or you'll be able to see it under the UV light. Okay, a method for characterizing spots on a plate, like if we want to talk about differences in spots and mixtures, right? We calculate what we call an RF value, which stands for retention factor. And that's calculated by measuring the distance from the starting line to the top of the spot, and then dividing that by the distance that the solvent traveled, right? So again, from the starting line all the way up to the solvent front. You can see that the blue spot, the highest point was 3.1 centimeters, solvent moved 8.5 centimeters. So when we calculate that out, we get an RF of 0.6, and the yellow spot has a higher RF of 0.72. So it's important to recognize the the relationship between an RF value and polarity. So the higher the RF value, the less polar the compound is, and the more highly it moves up the TLC plate because it doesn't have as great of an affinity for polar silica gel. So it's important, to try to use a pencil to mark the solvent front when you take the plate out of the developing chamber in order to make sure you have this line, right, to be able to get this measurement to then be able to accurately measure the RF value. And the solvent will evaporate. You need to mark it before the solvent evaporates, immediately after the plate comes out of the developing chamber. Okay, so this is a process. You see somebody is using a capillary tube. They've got a bunch of different mixtures that they're spotting onto the same plate. They drew a line with pencil. They mark where they're going to place all the different mixtures so that it's nicely labeled at the bottom. Although I did notice that it's a little funny. It goes 1, 2, 3, 4, 5, 6, 5, or maybe that's S. I don't know. Anyway, you mark it however you want, whatever makes sense to you. And they're going to place these mixtures onto the plate. And he's going to clean the capillary tube, he or she will clean the capillary tube in between each so that it's not contaminated with the previous sample when you're trying to place the next spot. And he's doing that with a little beaker full of acetone. So after he's spotted some on this spot number 2, he will touch the capillary tube, he or she. I keep saying he, but I don't mean to do that. He or she or anybody, they might take the capillary tube and come over here and blot it onto paper towel so that all the compound comes out. Then they'll rinse it with acetone, blot that onto the paper towel so that it's entirely clean for the next sample. And this is the process of cleaning the capillary tube in between spotting the different samples. Okay, we talked before about the fact that you can use TLC to sort of follow or monitor a reaction. Let's look at this picture to see sort of how that's being displayed. So what they're saying here is that X is the starting material and Y is the desired product. So they have standard samples of these that in this case, they've mixed the two so that we have the 2 spots. And then now they're looking at the reaction progressing over time. So before the reaction is even started, before they've added all the reagents, they take a TLC at what we call 0 hours, right? So the very beginning, that should just contain the starting material, right? We haven't formed any product before all the reagents have been added and the reaction has progressed. So after half an hour, they spot another sample of the reaction mixture onto the plate here. You can see that we're starting to form a little bit of product, but we still have the majority appears to still be the starting material. After an hour, it looks like there's more starting material than reaction mixture, or more product than starting material. And then after 2 hours, still the reaction is progressing. There's still starting material present, but after 5 hours, the reaction is done. And again, they said, Y is the desired product. So this is great. It looks like the reaction is done. There's no more starting material. If they don't continue, if they don't stop the reaction, let it continue, and take a TLC after 8 hours, sometimes it's possible that you will have a continued reaction where the product continues to react to form a different product. So this is presumably like an undesired product that's forming after a certain period of time, right? So you can see how TLC can really use to monitor the progress of a reaction, and it can help us determine the optimal time for a reaction to take place, right? So can you tell what is the optimum reaction time? It looks like, right, it's about 5 hours. Any longer than that, we run the risk of getting an undesired product forming. And any less than that, you might still have some starting material left over, which would mean there's a mixture that has to be separated. And so that's just not ideal for an organic chemist. Okay, so let's talk now about column chromatography in a little bit more detail. We use columns that can vary in size. This is just a Pasteur pipette column. You guys may remember using the glass Pasteur pipettes in the lab. You can actually use those as a tiny little column if you want to, but usually we use a column that's a little bit bigger with a stopcock in order to stop the solvent from coming out before we want it to. And then typically we would plug the bottom with a little bit of cotton. That will prevent what we put above it from coming out the column. We don't want to happen. We just want solvent and dissolved compound to come out the bottom of the column. Typically above the cotton, because this is kind of a bumpy layer of cotton, we'll put a layer of sand so that we can have a nice flat surface to have the bottom of the silica gel sit on. And then a silica gel slurry will be added to this in order to get the stationary phase into the column. The compound is placed at the top of the column, and the solvent or LUN is used to push the compound through the column until it comes out the bottom of the column. So how do you know what size column to use, right? So that's really important. Is a little Pasteur pipette column sufficient, or do you need a giant column, right? How do you know? So the important thing about the size of the column is that it's going to depend on the amount of the sample that's being purified. So if you have a smaller amount, you can usually get away with a smaller column. If you have a larger amount, like many grams, you might need a much larger column. But it's not just dependent on that, it's also dependent on the difference in RF values of the different components in the mixture. So for example, if you have two components in your mixture and the difference in RF value is pretty small, that's another reason to use a larger column. And if the difference in RF values is really large, it will be fairly easy to separate that on a smaller column. So that can, also be a factor. So more specifically, though, the diameter of the column should be related to the amount of the sample. We would typically want the compound to spread out in a thin layer at the top of the column and then move sort of uniformly through the column. If you put a lot of compound and the diameter is really small, it'll be a nice thick layer of compound. And then as it moves through the column, it kind of gets bigger and bigger. So you potentially run the risk of having less separation if the band of compound is thicker at the top. So it depends, again, on the amount that you have and the difficulty of the separation. The length of the column will really mostly depend on the difficulty of the separation. So if the compounds are close together, you need a longer column to allow for more chance for the compounds to separate before they elute out of the bottom of the column. Okay, so you've chosen the size of your column, and now we need to fill the column with the stationary phase. So I think the most efficient method is a wet packing method, also known as forming a silica gel slurry. There is such a thing as a dry packing method, but that usually doesn't give the best results, in my opinion. So we are going to learn how to make what's called a silica gel slurry. Kind of looks like the consistency of a Slurpee. To wet the silica gel in order to put it into the column. And that way it's always wet and it forms a nice homogeneous mixture for the stationary phase for the compounds to then separate through. And it's super important that we never let the silica gel get dry once it's in the column. It needs to always be wet with solvent. You always want to make sure and monitor the amount of solvent that's in there and never let it go below the top of the silica gel, that means that you can form cracks in the silica gel, not homogeneous anymore. Cracks are channels that compounds can sort of move through the column without actually binding to the silica gel. So it makes it much greater chance that your separation won't be ideal if those cracks form. Okay, once you have finished adding the silica gel, sometimes people put a layer of sand at the top. And again, I'll be demonstrating this entire process for you while I'm in the lab. And then they put the compound on the top, and the compound will move through the stationary phase and again come out the bottom. What solvent or eluent are we going to use in our column, right? We've chosen the size of the column, we've packed it with a silica gel slurry, right? And now we need to know, okay, what solvent or eluent am I going to use, right? Should I use dichloromethane, ethyl acetate, methanol, some sort of mixture of some of those, right? How do we know what to do? Well, usually the eluent is chosen by TLC analysis. So we want to do a TLC. Hopefully we can see some nice separation using that eluent, and then we'll be able to know that that would be a good solvent to use for column chromatography. Generally, the more polar the compound, the more polar the eluent needs to be. Otherwise, it'll just be stuck on the top of the column, bound to the silica gel, and it won't progress through to the bottom of the column. And if it doesn't come out the bottom of the column, you can't isolate that compound, right? And you just lost it to the silica gel, which is not good. So you can use just one solvent, like pure dichloromethane or something like that, or you can use a solvent mixture. The only thing is, if you use a solvent mixture, you need to use solvents that are miscible, right? So you can't just use two solvents where they're not going to mix together. That's why some solvents are more common than others, because there's miscible mixtures where one of the components is more polar than the other, and that helps us make different solvent mixtures with different polarity. For example, if I'm using ethyl acetate and hexane, ethyl acetate is much more polar than hexane. So I can have a gradient where I start off with pure hexane, or maybe 10% ethyl acetate and hexane. And then later on, I can increase to 20% ethyl acetate and hexane, and maybe 30% ethyl acetate and hexane. And you get the idea, right? Until all the compounds dilute out of the bottom of the column. Okay, when you are running the column, there's some important things to note and to remember. Again, you don't want the column to ever run dry. So eluent or solvent is continuously added to the top of the column as needed. The solvent you can allow the solvent to come out the bottom with the compound in it using gravity as your only method of getting things to flow out the bottom, right? Gravity will push the solvent through, but that's a pretty slow process. So oftentimes we use a gas, which could be air, or even better, an inert gas like nitrogen gas or argon or something like that, to push the solvent through the column. And that speeds up the column. It'll be much faster if you do things that way. And that process is called a flash column chromatography. So if you use a gas to help the mobile phase move through the solid phase more quickly, that's called a flash process. Okay, so at the bottom of the column, typically we're going to collect fractions. And if you can't see the compounds, like if you can't, because they're not colored, right? Then you just collect a set volume, like you fill up the test tube, and each test tube has 10 milliliters of solvent that it takes to fill it up. So you would just collect a set volume in every single fraction. If everything's colorless and you can't see what's going on. If you have two compounds and one's orange and one's blue, that's not a very common situation, but it's possible. Right? Then you could sort of watch the orange column move, the orange compound move through the column and eventually come out the bottom, and sort of collect that all in one giant Erlenmeyer class. And then once that's done, you can sort of then change to a different Erlenmeyer flask and use a different solvent to push that blue compound through, and then collect that a giant Erlenmeyer flask. But usually things are colorless, so we just kind of use smaller test tubes so that we can catch, the point where one compound comes out and the other compound then starts to elute, and you get a better chance of separation that way. Okay, once you have all these fractions, regardless if they're colored or not, you may want to do a TLC analysis to figure out what the heck is in each of those fractions, right? Which compound is in them. So we would do TLC on all of the fractions to visualize what's in those fractions. Sometimes nothing but solvent is in those fractions, like especially initially, before the compounds have had a chance to make it to the bottom of the column, very likely that the only thing coming out of that column is just solvent, and so there's nothing of interest in that particular fraction. That's, normal. So if you're doing TLC and you see the initial two fractions don't have anything in them, no spots, that's very normal, okay? Okay, so after we've done our TLC analysis and we've identified which fractions contain the same pure components of the mixture, we can combine those into a round bottom flask and use the rotary evaporator to remove the solvent. And then hopefully you have a substantial amount of a pure compound of interest. So this is just a visual, right, that kind of shows the process of the column. So this shows it where we've already made the column and we're adding there's LUN at the top, right? So there's always solvent. We never let the solvent run below the top of the column. And then, but eventually we'll drain the LUN just so that it's at the top, right here, right? So that everything is wet below it, but there's no solvent above it. That's when we're going to introduce the sample. So it's like these little squares are the sample, right? And the little circles. That's introduced to the top, right? And it goes into the column. And then as LUN is added and it moves to the column, the compounds then again separate based on their different polarity and different affinity for silica gel. We continue adding LUNs, right? And collecting fractions. And then eventually one compound would hopefully come out. We collect a fraction of that. And then we would, once that's all come out, we can hopefully switch to a different fraction, allow the second compound to come out. Again, there's always solvent being added on top to help push that through. And then the second compound elutes off of the column, and now you have two fractions that have separated components in them and several fractions that maybe just have solvent in them. So that would be an ideal separation. Okay, let's talk a little bit more specifically about the experiment. There are 4 parts to the experiment, and we do it over 3 days in the lab. In the first day, we're going to do parts A and B. And part A is using TLC to identify an unknown. So what we're going to do is you're going to get standard samples of fluorine, fluorinol, and fluorinone, all dissolved in an organic solvent. And so you'll come and get a standard sample for each of these 3. And then you'll get an unknown, right, which is one of these 3 compounds. And we'll spot that into a 4th lane. And also we can do a reference mixture, which should be given to you as well, where we've mixed A, B, and C. So we've spotted 5 different solutions onto one plate. And then we're going to allow those spots to move up the plate during the development process and visualize the spots after they have migrated up the plate. So let's say this is what it looks like after developing the TLC plate. Perhaps fluorine has moved up to here, right? Fluorinol is down here and fluorinone is here. And that would make sense because if you go back and look at the different functional groups, right, and you'll see how well each functional group is likely to interact with silica gel. And again, the less polar the compound is, the less likely it is going to adhere to polar silica gel, and the higher up the plate it will go. So this one being the most polar, it makes sense that it is the spot that has migrated the least and has the lowest RF value. Again, D was our unknown mixture, and then E was a reference mixture of all three. And that's just used so that we can actually see in this lane all three compounds separated. So then we're looking at the unknown and we say, okay, which one does it correspond to? Looks like it corresponds to spot B, so our unknown is fluoronol. So that's the goal of part A, to identify an unknown by TLC. And then after you do that, you're going to move on to part B of the experiment, where you're going to look at selecting the correct solvent for TLC. So when organic chemists are initially training on how to do all these techniques, they don't really have a good understanding of sort of which solvent to use for different compounds based on their polarity. So that sort of intuitive sense comes over time. But the pre-lab actually tries to get you to think about this a little bit by asking you to go back to a previous activity where we actually looked at dipole moment and polarity of these different solvents and sort of rank the solvents in terms of their polarity overall. So you can get an idea of the polarity of different solvents. So we're going to develop 3 identical plates using 3 different solvents, right? And for example, if we used hexane, ethyl acetate, and acetone, there's a manual, but this is like an example. So I guess we're doing these, but this is a little bit, this picture I took from somewhere else, where they actually used 100% hexane, 100% ethyl acetate, and then a 50-50 or 1-to-1 mixture of ethyl acetate and hexane. And then they're going to develop them with the 3 different eluents in 3 different developing chambers. And again, they have sort of a starting material spot, a product spot, and then a reaction mixture. In their particular case, in our case, we're just having standard compounds, so there's no reaction in this case. But anyway, regardless of what you're spotting on the plate, you then allow those to develop and see how far the spots move, right? This would be an example where the spots haven't moved enough to really be able to see very clearly the difference between the starting material and the product. So this eluent is not ideal for separating these components on a TLC plate because it's too nonpolar, right? And therefore the compounds have really just adhered to silica gel, and the solvent wasn't polar enough to disrupt those interactions and move the compounds up the plate. What about ethyl acetate? 100% ethyl acetate made all of the compounds move to the top of the plate. Again, that's not ideal for seeing the best level of separation that we possibly can. Instead, this 1-to-1 ethyl acetate hexane mixture for the eluent was perfect, and it allowed us to see some great separation. So ideally, the spots are somewhere between like 0.2 and 0.8 in RF, and you can clearly differentiate different components in the mixture. That's an eluent with solvent that's just right in terms of the choice. So you're going to look, and in part B, using the same compounds, run them with different eluents and see, right, what would be just right and what's maybe too polar or not polar enough. On the second day that we come into the lab, you guys are going to do part C, which is monitoring a reaction by TLC. The reaction that you're going to do is the conversion of fluoronone to fluoronol. We kind of chose these for a good reason. Again, these are highly conjugated aromatic compounds, which means that they show up nicely under ultraviolet light, so we're easily able to visualize the spots. But in addition, we use these same compounds in parts A and B of the experiment, so you're going to be really comfortable with these spots and know what solvent to use to develop the plate as you're monitoring the reaction. You'll almost have optimized that in the previous part of the experiment. And then this reaction is specifically a reduction of a ketone to an alcohol. Now, we haven't covered reduction reactions in the lecture component of this course yet. Typically, that's covered at the very beginning of the second semester of organic chemistry. But you don't really need to have looked at the reaction before to just note that we are going to convert a ketone to an alcohol through the use of sodium borohydride as a reagent and methanol as the solvent for the reaction. Another reason why this reaction was chosen is because the reaction is very quick. We wouldn't want to choose a reaction that takes 5 hours to complete, since you need to do it in 3 hours, right, while you're in the lab. And we're, constrained on time. So the reaction is actually typically over, in just a few minutes. So that works really well. So we can, take multiple samples at different time points and really see a quick progression of the reactant to the product. So you're going to be spotting a standard and the starting material in its own lane. And then you'll be taking samples at different time points once you've added the sodium borohydride to the mixture of fluorinone and methanol. So before you add sodium hydride, that's your 0 time point. And that should just have this spot in it by itself. And then once you add the sodium borohydride and the reduction reaction starts happening, we're going to see a conversion of fluorinone to fluorinol over several minutes. And so we'll take samples out at different times. So you need to have a stopwatch ready or your watch ready or your phone, and make sure you're really paying attention to get the samples at the exact right time. When you spot them onto the plate, that should be at the time that you're trying to do. So for example, if you're doing 30 seconds, you want to get a sample from the reaction mixture at about 27 seconds, and then have it spotted onto the plate at 30 seconds. Because once you've spotted onto the plate, the reaction mixture is quenched or stopped. So we're going to see exactly what's in that mixture at that time point. And then after several minutes, hopefully the reaction is over. And if it isn't, then you're going to double the amount of sodium borohydride and try it again and see that it, by adding more reagent, it's going to speed the reaction up. And we'll be able to see that play out through the TLC process. So one thing to note, right, is it kind of makes sense that as our product is more polar than the starting material, alcohols are more polar than ketones, then we're expecting the product spot to have a lower RF than the reactant spot. So this is something that would make sense for a complete reaction or conversion of fluorinone to fluorinol. But this isn't one that has looked at several different time points, so yours will look a little bit different. Okay, moving on to part D of the experiment, we're going to do our first column chromatography. And we're going to try to separate fluorinol and fluorine. And you can do a TLC of the mixture to see what it looks like, right? With the 2 components in there initially. And then I'll show you how to set up a column and how to put the compound in at the top, and then solvent in, and use air pressure or nitrogen gas pressure to move the solvent through the silica gel. In this series, which I stole from the web, you have 2 different compounds that you can see, right? It's sort of this red compound and the orange compound, and you can see them moving through the column over time. In our case, one of these compounds is yellow and the other one is colorless. So we will not necessarily be able to visualize both components migrating through the column because one of them is colorless. So that kind of simulates a more realistic environment anyway. Usually things are not super well colored in organic chemistry. Okay, and hopefully we get the 2 components separated at the end of the process. We'll know if we do by TLC analysis. So again, we're going to collect a series of fractions. Some students like to number their test tubes to make sure that they all will remember the order. Other times, students will just decide on a pattern. So maybe they start in the lower left and then move this way, and they do like a snake pattern. So they'll just know in what order they're going to use them without numbering, but it's up to you and your comfort level. Then all of the fractions, once they've been collected, again, will be analyzed by TLC, and we'll be able to see if the components were effectively separated. And you'll note that in your data sheet. Okay, so moving on from talking about this particular experiment, before we go, because it's a type of chromatography, I wanted to take a brief look at gas chromatography. We did use gas chromatography in a previous experiment. If you will recall, when you were separating cyclohexane and toluene in the distillation experiment, you took your fractions that you collected from distillation and put them in little vials, and the lab tech analyzed them by gas chromatography to let you know what was present in each of those fractions. And gas chromatography is a much more quantitative process compared to, for example, TLC, because it will it'll allow you to look at exactly how much, right, of each component is in there percentage-wise. So that's great advantage for gas chromatography. Okay, let's look at a little bit about what's happening in the instrument. So with gas chromatography, we typically have a steel column, and it's placed in an oven in order to maintain a uniform temperature. And interestingly, in this type of chromatography, it's a little bit different from TLC and column chromatography in that the stationary phase is not a solid, it's a liquid. And the mobile phase is not a liquid, but it's a gas, right? So that's a little bit different from our previous types of chromatography. And the again, the stationary phase is a liquid, and it's typically a high boiling organic compound. High boiling means it has a high boiling point, and it's absorbed onto the solid inert packing in the column. Inert means it doesn't chemically interact with the compounds. And again, the column is usually steel, but it can be made of other materials. And again, it's placed in an oven for temperature control. So you can see here, this is the mobile phase, it's the carrier gas. Typically, it's an inert gas, such as helium or argon or nitrogen gas. Those are common gases that are used in column chromatography. So you've seen those big steel tanks, right, that are right next to the instrument. Those are supplying the gas into the instrument. And then there's a flow controller. There's a point where you can inject a sample into the instrument, and it gets inserted into a flow of the carrier gas and gets vaporized. And so the separation is based on both the ability for the compounds to vaporize and their interaction with the liquid stationary phase. They're actually not interacting with the mobile phase or the gas, because again, it's an inert gas. So the gas is just used to kind of push the compound through the column, and then it's based on its different adherence to the high boiling organic liquid in the column. Eventually, it reaches some sort of detector, and that could be a mass spectroscopy detector, where compounds are ionized, and then we identify what the compound is by its mass. And again, mass spectrometry is not something we've talked about yet. That'll be talked about more in Organic Chemistry 2 lab. But something will detect the compounds as they come out of the column. And there's different types of detectors, not just using mass spectrometry. And then eventually, the detector will create a spectrum for us where we can look at different compounds as they elute. And this is, a giant mixture, but typically, if there was only one, then it would be pure, just one compound, right? So then we can see not only how many components are in the mixture, but exactly what percentage from each one, which is obviously an advantage to TLC, but the advantage to TLC over gas chromatography is TLC doesn't require an expensive instrument, relatively cheap and fast to perform. Disadvantage is its qualitative analysis as opposed to quantitative analysis. All right, hopefully you learned a lot from the pre-lab lecture, and I will be sure to demonstrate and show you in the lab with the glassware and things exactly what to do. So I look forward to seeing you soon in the lab.